Good afternoon, everyone. I'm Rick Lifton, president of the Rockefeller University, and I'm delighted to welcome you back to another installment of our webinar series, Virtual Discussions with Genuine Experts. Today's webinar is our second session this fall, and I'm pleased that the focus of our discussion will be the exciting findings that have come from research whose goal and approach was described to you last spring. Since that time, the idea and execution on this project has been spectacular, and it's my great pleasure to welcome back my colleague, Dr. Jean-Laurent Casanova, who brings us the latest news from the COVID human genetic effort. As we all know, the world has been watching anxiously, hoping for scientific breakthroughs to combat COVID-19 and wishing for them to occur in record time. You'll recall that when Jean-Laurent told you about his international collaboration last May, he and colleagues were in the process of collecting and analyzing DNA from patients from around the world to understand why, while most patients with COVID-19 infection have mild to moderate courses, with some individuals remaining asymptomatic, others have progressive, life-threatening disease. Jean-Laurent's project sought to identify mutations in the genomes of affected patients that make them vulnerable to severe outcomes with COVID-19 infection. It was proposed that success with this approach might further provide insight into why other patients without these DNA changes go on to severe disease. Today, he will tell us of the remarkable progress his team has made. As we've discussed previously in this forum, our scientific community has united to battle COVID-19, part of a global effort bringing together academic institutions, biotechnology and pharmaceutical companies, and governments to advance this work as quickly as possible. At the same time, we've seen an equally remarkable outpouring of philanthropic support for our COVID projects. I want to thank all of you who have contributed to this effort. Your support has been vital to the work that you will hear about today and to all the scientists at Rockefeller, like Jean-Laurent and his lab, who took on COVID-19 and have been working feverishly, figuratively speaking, to achieve their terrific results. Before we hear from Jean-Laurent, I want to give a brief update on the COVID-19 pandemic. Starting here, we can see that on the top panel, this is the global number of new cases per day. And you can see that from where we were in September, the pace has continued to go up, approaching 400,000 cases daily on a global basis. This remarkably high number of cases daily has been accompanied by a sustained high death rate across the world, with a reported 5,000 deaths occurring every day attributable to COVID-19. This shows the global map of the incidence of disease as of our last meeting in September. And as you can see from this map, things were quite hot in South America, North America, part of Europe, and there was striking lack of activity in Southeast Asia and Sub-Saharan Africa. Switching to today, we can see that the map has changed significantly. The color of the United States is a deeper red than before, with an increase in the number of cases since we last met. Parts of South America have cooled off just a little bit. Russia has had a striking increase in new cases, and most notably for its change, Europe has become almost uniformly with high levels of new cases every day, with dramatic increases in the incidence of new cases in the UK and many other countries across Europe. So this shows the daily new diagnoses of new cases and deaths from COVID-19 in the United States since the start of the pandemic. And as you can see, over the last month, there has been a sustained increase in the number of daily cases shown in the top panel. We've gone from a trough of about 35 to 40,000 cases per day to a current average that's approaching 60,000 cases per day, with the daily incidence numbers in the middle of the last week approaching 70,000 cases per day, numbers not seen since July. This obviously is very worrisome. The current total in the United States is about 8.5 million cases since the onset of the pandemic. The bottom panel shows that there's been a sustained high rate of death in the United States on a daily basis, with the 7-day rolling average of about 800 deaths per day from COVID-19, totaling 220,000 deaths since the start of the pandemic. The distribution of cases across the United States is not even. There are some states that are achieving very high levels and other states that have relatively low levels of incidence right now. Most strikingly, when we met before the summer, there were very high cases emerging across the South. And this has substantially shifted now to the Midwest and the Mountain West. And you can see in the top part of the panel, the high prevalence of cases in North Dakota, South Dakota, Montana, Wisconsin, Nebraska, Idaho, Utah, Wyoming, and Iowa. Our territory, Guam, also has a record high number of cases, whereas states at the bottom two panels have quite low levels of new cases on a daily basis, including New York as being at a persistently low level compared to most of our other states in the United States. This shows the new cases per 100,000 population and the percent change over the last 7 days across the United States. And you can see this striking variability. So North Dakota, the last 7 days had averaged 652 new cases per 100,000 population. That's almost 1% in the last week have been infected. In contrast, if you look on the right side down toward the bottom, you can see this is in descending order. New York has less than 1/10 as many cases per 100,000 population, with just a very small increase in the number of new cases over the last week. So the rate of new cases has gone up modestly in New York, but quite strikingly in North Dakota, which has increased 27% over the last week. In fact, 39 states have seen an increase in the number of new cases per 100,000 over the last 7 days. And in many cases, this has been quite a striking increase, greater than 20% in 16 states. This is accompanied by an increase in the percent of COVID tests that are positive across the states. As you can see in this schematic of the country, the Northeast continues to have relatively low rates of positive tests, virtually all less than 5% of all tests being positive. New York continues to be quite low, an average of about 1.2% over the last week, 1.3% today. But other states have quite high levels. In the yellow color is where states that are between 5 and 10% positive rates, which is a very substantial fraction of the country, and in varying shades of deepening orange, increasing rates of positive tests, going from about 11% in Florida to over 15% in Wisconsin and Wyoming, over 20% in Idaho, and over 25% in Nevada, South Dakota, and Iowa. These are very high rates of positive tests and are consistent with the virus being well out of control across much of the country in ways that harken back. We haven't seen positivity rates like these top levels since the onset of the pandemic in New York back in March. Getting back to New York City, this shows the curves for number of cases, number of hospitalizations, and number of deaths attributable to COVID-19 every day. As you can see on the upper left panel, in the last 4 weeks, there has been an uptick in the number of new cases per day in New York, in the city, roughly doubling. And the city and state have done a good job of tracing the origin of this increase. And this is a modest number of zip codes in the city. About 20 out of the 146 zip codes in the city have seen significant increases in the number of new cases and the rate of positivity that has in some cases well exceeded 5%. I was very concerned 4 weeks ago that this was going to result in explosive growth of new cases across the city. That has not happened as yet. You can see that the rate increased to a peak about 2 to 3 weeks ago, and has been coming down slightly, and at worst remaining steady since that time. It's been accompanied by a parallel increase in new hospitalizations, 40 to 55 new hospitalizations across the city on a daily basis. To date, we haven't seen an increase in deaths in the city, but that almost invariably follows about 3 weeks behind in increases in new cases. So I fear that we will see a parallel increase in new deaths coming on the horizon. This is a relatively precarious position. It's been apparent that the city and state have done very good jobs of intervening in these communities where new cases have been clustered, and hopefully this will attenuate the spread of the virus and we'll be able to tamp this down. It's, I think, very gratifying that we have not seen the explosive growth that we've seen across much of the rest of the country. Over the last month, a few new things have occurred that I think are worth reporting, among many that we could talk about. We've seen the first data in humans using neutralizing monoclonal antibodies. You've heard a lot about the monoclonal antibodies that have come from Regeneron. And in fact, Regeneron has been used as shorthand for these new therapeutics that are in clinical trials. They seem to be capable of knocking down viral levels in patients who have not started making their own antibodies as yet. This may be reducing the progression of disease in these patients, but we're early days, but I'm aware that data has been submitted to the FDA for consideration for an emergency use authorization. This is very promising. Cautionary note, monoclonal antibodies are not going to be available in the numbers necessary to treat everyone around the world. These reagents can be made in the millions, not the billions at best, and will be in limited supply even in their optimum use. They also do not last forever, so they need to be given repeatedly. They are a parallel treatment for patients who either do not respond to vaccines or are at high risk, who may not have yet received a vaccine. Plans for vaccines are moving rapidly along. We heard about these from John Bell last month. Since then, the companies that have vaccines in trial have talked about their plans for registering their vaccines. Most of these trials include the registration and an engagement of 30,000 to 60,000 individuals. Some are including elderly people in these trials, some less so. Children have not been typically included, but there are plans afoot to include children in trials, so that we will know soon whether vaccines that work in adults also work in children, which would be expected. The biggest issue for vaccines will not be proving whether they work or not, but proving that they are safe. And if they have side effects, how frequent and severe those side effects will be. And that's why these trials need to be relatively large. I should point out that if the trials are being conducted in a place where there's a lot of active infection, for example, in the Dakotas, where I mentioned that nearly 1 in 1,000 people will become infected every week, you could get a lot of cases very rapidly in a 30,000 patient trial. Sufficiently so to demonstrate efficacy if with 100 cases with the expectation that you'd have 50 in the placebo arm, 50 in the vaccine arm. If you had a vaccine that was 80% effective, you would see 50 in the vaccine arm and only 10 in the vaccinated arm. That would be very powerful evidence that the vaccine was working. And the power calculations have been that you want the 95% confidence interval to assure that the vaccine is at least 50% effective in preventing infection. So you need a number well north of that if the numbers are small to be assured that the vaccine is effective. Proving safety is another matter, and for this reason, it has been proposed by the FDA and accepted by all of the companies, to my knowledge, that there will need to be 2-month follow-up on patients who have received the active vaccine to have an assessment of their safety before they will be considered for registration. Seems likely that these will hit the finish line by the end of the year in many cases. However, several of these vaccine candidates are on hold with the FDA right now, owing to side effects that have occurred typically in a single patient who may have a vaccine-related side effect. But we need to accumulate a lot more data before we understand what the spectrum of side effects might be. The second major advance has been evidence that interferon deficiency strongly predisposes to severe COVID-19. This is the topic of today's webinar, and I won't elaborate on that further. And then lastly, we've heard renewed discussion about herd immunity, that maybe we should just stop protecting everybody but individuals who are strongly predisposed to adverse outcome from COVID-19 and let the disease take its course in the general population. And I just want to give a couple of comments on the notion of letting herd immunity become our approach. This would inevitably require us to achieve herd immunity to have many more people infected than we do now. So the development of herd immunity depends on how infectious the virus is. And I should point out before getting there that it's been said that, well, herd immunity, we need to protect the elderly so that they don't get infected and prevent them from having adverse consequences of infection. And it's worth pointing out that when you look at the age distribution of people who have died from COVID-19, it is undoubtedly the case that about half of people who have died have been over the age of 75. But that also means that half are under the age of 75, and 75 is at the low end of life expectancy in the United States these days. Average life expectancy for men and women is about 80 today. And as a consequence, as you can see, there are many deaths under age 65, under age 55, and thousands still under age 45. And for this reason, because many of these individuals also have underlying susceptibilities, which we'll hear more about this afternoon, it is very difficult to define, we'll just protect the elderly. Where should we draw the line on the elderly today? And who is going to be willing to be part of the group that we just say, well, we try to protect everybody who is at risk. There are many people who are at risk, as evidence from the data that we have in hand today. So to achieve herd immunity, there have been suggestions that maybe with 20 to 30% of the population infected, we could achieve herd immunity. I don't think anybody credibly believes that number to be accurate. The level of immunity that you need to achieve herd immunity, it depends on how infectious the virus is. This shows the R0, the number of people who typically get infected from one infected individual without any interventions. And you can see at the top is measles, which is extraordinarily infected. At the bottom is influenza, which is not highly infectious. And for that reason, we can do quite well with vaccination for influenza, even if we only get a third to a half of the population to be vaccinated with an immune response. COVID-19 is more infectious than influenza, and it's, I think all credible estimates are that we need to get to about 60 to 75% of the population immune before we will have so-called herd immunity. It's worth pointing out that herd immunity does not mean that nobody gets infected after that. It just means that infections are not typically going to be able to spread very far into a community without breaking the chain of transmission, without any specific intervention. Because once that high proportion is infected, the virus will not find as frequently people who are still able to be infected, and local infectious cases tend to die out very quickly. So where are we now? A study just came out last month in The Lancet, actually a few days after our last webinar, that reported in a national survey of the prevalence of antibodies against the SARS-CoV-2 virus in the United States from a national sample. The sample was taken in July, and the prevalence of antibodies against the virus at that time was about 9% when you try to adjust for across the population of the United States. That level of antibodies ranged from low levels in the West, 3.8% in California, up to 33% in New York. So there's quite a gradient across the country. However, nobody is approaching the levels needed to achieve herd immunity. And going from a national level of about 9%, and even imagining that we're up to 20% today, we would still need at least a threefold increase, a tripling of the number of cases nationally to begin to imagine that we're achieving herd immunity. The number of deaths that would likely attend that dramatic increase would probably add another 400 to 600,000 new deaths to our roll. And I think most people would find that an unattractive outcome. So moving to what is going on in Rockefeller Laboratories, there's a wide range of projects ranging from convalescent plasma to neutralizing antibodies to the spike protein to the topic of today's work that Jean-Laurent will tell us about, to host genes required for the viral life cycle, which is a project that our Nobel laureate Charlie Rice has pioneered, and a variety of additional projects involved in drug screening and others that are well underway. And this remains a major area of interest across the campus. And I'm extremely proud of all of the work that our faculty have done. So with that, I want to back up and just remind you that 2 weeks ago here on campus, we had the remarkable morning where we woke up to the incredible news that our Rockefeller colleague, Charlie Rice, had been awarded the Nobel Prize in Medicine for his transformational work on hepatitis C virus. The hepatitis C virus was discovered in 1989 and was found to be the cause of about 400,000 deaths annually from liver failure around the world. However, from the time of its discovery in 1989, for the next 7 years, efforts to propagate the virus or to get it to replicate in cells failed, thwarting efforts to develop screens to enable discovery of new drugs that would prevent progression of liver disease. A hardcore basic science virologist, Charlie had the seminal insight from his pioneering work on yellow fever virus that the then known genome of the related hepatitis C virus was incomplete, missing a short piece at the end of its genome that Charlie deduced was essential for viral replication. So Charlie not only proved that this was correct, he went on to make a miniature version of the viral genome that was non-infectious, but perfect for screening for drugs that inhibit the viral life cycle. This led to the development of highly potent and extremely safe drugs that remarkably can cure nearly all people who have chronic infection with hepatitis C. These drugs have already been given to more than 5 million people, and they will continue to prevent disease and save millions of lives. It's an incredible demonstration of the power of science to improve the human condition. I should add that it's no surprise that Charlie is among the Rockefeller faculty now making vital contributions to COVID-19 research. This includes his collaborative work with Jean-Laurent, who you will hear from. So now it's my great pleasure to introduce my colleague, Jean-Laurent Casanova. Jean-Laurent is the pioneer in the study of genetic predisposition to infectious diseases. Jean-Laurent is head of the St. Giles Laboratory of Human Genetics of Infectious Disease at Rockefeller and an investigator of the Howard Hughes Medical Institute. He's an elected member of the National Academy of Sciences and the National Academy of Medicine. Jean-Laurent's work has shown that individuals can be susceptible to severe outcomes from common infections simply because they have inherited a mutation in a single gene. In recent years, he's uncovered single gene mutations that disable immune defenses against tuberculosis, invasive pneumococcal infection, and the devastating brain infection herpes encephalitis. His findings have led to treatments that have saved many lives. In our virtual discussion webinar in May, Jean-Laurent described the efforts he was undertaking to understand why the novel coronavirus leads to severe outcomes in only a fraction of those who become infected with the virus. He had already launched the COVID human genetic effort, an international initiative that's been collecting patients who have been infected and determining their genome sequences and clinical outcomes from thousands of patients across the globe. The scientific objective was to discover mutations that might render patients vulnerable to severe COVID-19. Jean-Laurent and his colleagues have delivered on that objective in record time. In back-to-back papers in the journal Science, they reported on 2 sets of host factors, aberrant genes and misguided antibodies that account for severe disease in a significant number of severely ill patients. This rapid progress is nothing short of remarkable. So I'm delighted to now turn the screen over to Jean-Laurent to share his fantastic story with us. And at the end of his presentation, we will answer viewer questions. You're welcome to submit questions at any time using the Q&A function at the bottom of your screen. And now let's hear from Jean-Laurent.
Thank you very much, Rick. I want to thank you for inviting me to give this webinar and also for having supported our effort from the first days onward. We would not have achieved these discoveries without your support. I will share with you, as Rick just said, 2 papers, the preprint versions of which were posted online by the journal Science a couple of weeks ago, but the final versions of which will be printed and posted online on Thursday in a couple of days. On May 7, when I gave a webinar, I told you about our hopes, our ideas, our plans, essentially our hypothesis. And I'm especially happy today to share with you the results of this endeavor that has kept us busy, us because we've worked hard in the lab and we've teamed up with a great number of colleagues, scientists and physicians worldwide, together in the form of a consortium, the COVID human genetic effort. So let's start with the beginning. And the beginning in science is always the problem posed. And the problem with which you're all familiar, but which I think we should briefly discuss, is that of inter-individual clinical variability in the course of SARS-CoV-2 infection. On the left panel, you have a representation of the problem. The vast majority of infected individuals do not even notice they're infected. The infection is silent. That's at least 90% of infected individuals. A smaller proportion, maybe around 10% of cases, will have an influenza-like illness. They will be sick, they'll stay at home, they do not need to be hospitalized. In less than 1% of the cases, the infection is severe and the patient needs to be hospitalized. In an even smaller proportion of cases, less than 0.1%, the infection in the form of a pneumonia leads to respiratory distress, and these patients are typically admitted to an intensive care unit where they are mechanically ventilated. And these infections are truly life-threatening. In 10 to 20% of the cases, unfortunately, they're lethal. So the problem is really, why would 1 in 1,000 or 1 in 10,000 infected individuals end up in an intensive care unit when others control the virus much better? So the epidemiologists have early on in March and April identified a major risk factor, which is age. Clearly, above the age of 45 or 50 years, there's a sharp increase in risk decade by decade. The risk of life-threatening COVID-19 pneumonia by the age of 20 or 25 years is maybe 1 in 10,000, whereas by the age of 85 or 90, it's closer to 1%, if not 10%. So age is really the major risk factor identified. Sex is also a risk factor, but to a much lesser degree. Women are protected when compared with men, but the magnitude of this risk factor is much less than that of age. And a number of comorbidities have also been proposed. Again, having diabetes or being obese or having high blood pressure does not help, but the impact of these comorbidities is relatively modest. So if you consider any demographic or medical category, still there remains huge inter-individual variability. There are sadly young women in their teens or 20s who died of COVID. And there are men age 100 or 105 years who handled the virus in a completely silent manner. So the fundamental question is, what is the cause of lethal or life-threatening COVID-19 pneumonia? That's the problem we've been interested in from day one. Once we have a problem, we needed a hypothesis. That's the hypothesis I discussed with you on May 9th. It is the idea that some patients with life-threatening COVID-19 pneumonia may be sick because from birth they carried DNA variations. In the past, people used to say mutations. We now say variants that make them vulnerable to SARS-CoV-2 infection. This idea didn't come up during an epiphany. It was based, as Rick alluded to work from our lab and other labs over the last 25 years that had revealed that for a great many viral, bacterial, fungal, or parasitic infections, striking previously healthy children, adolescents, or adults, the catastrophic disease could be caused not only by the infectious agent, but also by germline genetic variants that made the patients vulnerable to that particular microbe. So we decided to test this hypothesis in patients with life-threatening COVID-19. Importantly, the idea behind this project is not only that the discovery of these single gene inborn errors of immunity could be illuminating in patients bearing these mutations, but also that they would perhaps reveal mechanisms that could be disrupted by other causes in other patients who do not carry the single gene inborn errors of immunity. And as you will hear, it turned out to be the case for COVID-19. So we had a problem, a hypothesis. The third step, as always in science, is the approach, the plan. So early on, we thought that if there were genetic cases underlying severe COVID-19, they would display what in our jargon we call genetic heterogeneity. That is, patients would be mutated, each in a different gene, or at least that there would be many, many genes mutated, not in individual patients, but at the population level. And therefore, for example, if there is a patient in Brooklyn mutated in a gene, it may be that the other patient on the planet mutated in the same gene, maybe in Tokyo and the third in Jakarta. Therefore, we teamed up with thousands of hospitals and decided to follow the epidemic from east to west and enroll as many patients with life-threatening COVID-19 pneumonia as possible. These hospitals send the clinical information and ship a blood sample of their patients to sequencing hubs. And the sequencing hubs shown on the slide here, which are located in Oceania, Asia, the Middle East, Europe, Latin, and North America, these sequencing hubs sequence the genome of the patients. And then in our lab, we analyze all these genetic data transmitted by the 50 and more sequencing hubs. We've not only recruited patients in the intensive care unit with life-threatening COVID-19 pneumonia, but importantly, as controls, a group of subjects who have been infected with SARS-CoV-2, but who remained asymptomatic. So by October 15, I think we've already sequenced the genome collectively of 3,500 subjects. Maybe 2/3 of them had critical COVID-19 and 1/3 had remained asymptomatic in the course of SARS-CoV-2 infection. So we have a problem, a hypothesis, a plan, but as you see, this is a general plan and science needs to go by specific plans. The cohort I show you here is the cohort of patients on which we tested our first specific hypothesis. You see that the asymptomatic individuals on the left tend to be younger than patients with life-threatening disease. There are also more women among the asymptomatic than the life-threatening cases. On the right-hand side, you see a genetic analysis confirming that the international nature of our recruitment of patients and controls corresponds to also a great level of genetic diversity. We have patients of Asian ancestry, East Asian, South Asian. We have patients from the Middle East, Northern Africa, Europe, and both Northern and Southern Latin America. Now, in these cohort of patients shown here, about 500 controls and 659 patients with critical disease, we began by testing one specific hypothesis. And the idea is shown here. I know this slide is complicated, but I think I've shown you a slide resembling this one on May 7th. Let me try to explain. The hypothesis we first tested is that the genes that we had shown over the last 7 years, when mutated to cause life-threatening forms of influenza, that the very same genes, when mutated, could also be responsible for life-threatening COVID-19 pneumonia. That's the hypothesis. And what you see on the graph here are two types of cells. The plasmacytoid dendritic cells, which are leukocytes circulating in the blood, that have the particularity of being the cells that produce the greatest amounts of type 1 interferons in the body. And the key word is really here type 1 interferon. The type 1 interferons are a set of 17 molecules that are produced by these plasmacytoid dendritic cells, but also by all cells of the body, and that are potent antiviral molecules. They activate through their receptors and a signaling pathway that had actually been deciphered here at Rockefeller by Jim Darnell. They activate a number of genes that will protect cells from the body from viral infection or viral growth. So the plasmacytoid dendritic cells are in the blood, and I've also represented a larger cell, which is one of the many epithelial cells in the lungs. In these cells, the virus can be recognized by a molecule called toll-like receptor 3, which via another molecule called RF7 is going to result in the production of type 1 interferons. So both the plasmacytoid dendritic cells in the blood and the cells of the lungs, epithelial and other types of cells, make interferon, and the molecules in red shown here, when mutated, are responsible for severe influenza pneumonia. The molecules shown in blue are molecules encoded by genes, the mutations of which we had shown in the past to be responsible not for influenza, but for other viral illnesses. So by inference, we considered these 13 genes controlling the production and amplification of antiviral type 1 interferons in leukocytes in the blood and in the lungs as candidate genes. And therefore, we analyzed variants at all these loci in the cohort of patients and controls that I've described to you. And I'm not going to bombard you with the biochemical or immunological analysis of all the variants at these loci. I summarize in that slide the key finding in this first study. In 23 patients, that is approximately 3% of this international cohort, we've discovered what we call autosomal dominant or autosomal recessive disorders. Autosomal dominant disorders mean that the patients carry one mutation. Each gene is present in 2 copies in the human genome. And the autosomal recessive disorders are characterized by 2 mutations being present in the patient. There are men and women. The age of the patients ranges from 17 to 77 years. And you see from the column indicating their ancestry and residence that really the recruitment of patients was international. Most patients have survived. 2 of the 23 patients died. The reason that I've colored in red the autosomal recessive disorders is because these 4 patients lack the corresponding protein, RF7 for 2 patients and IFNAR1 for 2 patients. And RF7 is the gene that controls the production of all type 1 interferons, whereas IFNAR1 is the gene that controls the response to all type 1 interferons. So what we learned from these patients, age 49, 50, 38, and 26 years of age, who had never prior to COVID-19 been hospitalized for any severe viral illness, what we've learned is that very surprisingly from an immunological standpoint, these molecules were redundant for immunity against most common viruses. Until they were infected with SARS-CoV-2, these patients were fine. And that had implications that we're going to discuss in a moment when we decided to transition from the study of inborn errors to the potential discovery of other mechanisms in patients not carrying inborn errors. So this first study showed that about 3% of our international cohort of patients with life-threatening COVID-19 pneumonia were sick because at birth they carried variants in genes controlling type 1 interferon immunity. The clinical implications are simple to understand. Someone whose genome or exome, that is the sequence of the genes in the genome, is known, can find out if he or she is predisposed to severe COVID. If this is the case, it turns out that 2 of the 17 type 1 interferons are commercially available, beta interferon, that is used, for example, in MS, and alpha interferon, that was used for a long period in patients with hepatitis C virus, hep C virus hepatitis. Now, these findings pinpoint a mechanism at play in about 3% of patients, and there are many more genes revolving around type 1 interferons that we currently study. But as I alluded to earlier, in our mind, genetics operates like a compass. It is giving you a direction. And the direction here, as you've understood, is type 1 interferon. So we hypothesize that perhaps in other patients, there could be mechanisms disrupting type 1 interferon production or activity different from the inborn errors that I've just discussed. And we focused our attention on a possibility, a hypothesis, that is that the antibodies, which you've heard can be directed typically against SARS-CoV-2 or other infectious agents, well, that perhaps in some patients, there would be antibodies directed against self, what we call in our jargon auto-antibodies, responsible for autoimmune conditions, that perhaps there would be auto-antibodies against the type 1 interferons themselves, resulting in vulnerability to SARS-CoV-2 infection and disease. Why this idea? Well, first of all, as you heard, because we had discovered inborn errors of type 1 interferon, incriminating the disruption of this arm of immunity in patients with severe COVID. But second, because the autoantibodies against type 1 interferons had been described from the 80s onward. That is, over the last 40 years, a variety of patients have been found to have autoantibodies against type 1 interferons, but they were silent. For example, patients receiving alpha interferon or beta interferon as a therapy sometimes develop autoantibodies against interferon, but they've never been noticed to have, particularly severe or unusual viral illnesses. Also, a small minority of women with lupus, systemic lupus erythematosus, an autoimmune condition, have these autoantibodies. And there's also a rare disease, a monogenic form of autoimmunity. I'm not going to get into the details, where all patients with this condition have the autoantibodies against type 1 interferon. But in these three groups of patients, these autoantibodies, which, as I've said, had been known for 40 years, had never been found to be causal or even associated with any particular viral illness. There is only one paper in 1984 by the late Ian Gresser, who had contributed so much to the field of type 1 interferon biology and medicine. He had reported a 60-year-old patient with severe chickenpox, severe zoster, and autoantibodies against type 1 interferon. So the eureka moment came when we found 3 patients with this rare monogenic form of autoimmunity that I discussed earlier, who were admitted to an intensive care unit. We immediately thought that perhaps the autoantibodies against type 1 interferon were at play. And this slide here is, I think, unquestionably the most important slide of the seminar, the webinar today. We tested 987 patients with life-threatening COVID, and in 101, we found autoantibodies, more than 10%. In contrast, in subject with an asymptomatic infection, 663, we found none. You don't need statistics to see that this is highly significant. In the general population, a group of 1,224 healthy controls sampled prior to the COVID pandemic, we found 4 positive individuals, approximately 0.3%. So clearly, these autoantibodies are present in patients with life-threatening COVID-19 pneumonia in much greater proportion than in other groups. Importantly, as indicated in the bottom of the slide here, 95 of the 101 patients with autoantibodies to type 1 interferon and critical COVID are men. And half of them are older than 65 years of age. Therefore, these autoantibodies to type 1 interferon, which we have shown pre-exist SARS-CoV-2 infection, are in much greater proportion in men than women, and also increase in frequency with age. We've recently teamed up with the New York Blood Bank and also a blood bank in Finland, Denmark, and France to try to estimate more accurately the prevalence of these autoantibodies in the general population, decade by decade and by sex. What are these autoantibodies? Well, they recognize, as I said, the type 1 interferons. And as I alluded to earlier, there are 17 different related type 1 interferons. Their biochemical tree is represented on the left here. There are 13 interferons that are alpha interferons. There's an omega in blue, and in green, beta, kappa, and epsilon. What you see on the right-hand side here is the reactivity of plasma from patients against these individual interferons. And the observation, all the pink or red squares indicate positivity. What you see is that the patients with critical COVID-19 have autoantibodies against alphas, the 13 alphas, and omega, and more rarely against beta, kappa, and epsilon. It turns out that the alphas and the omega are structurally related, but also that they are the interferons that circulate in the bloodstream and circulate throughout the body. Whereas kappa interferon is more a skin interferon, epsilon, and the beta interferon tends to stay in the close vicinity of cells that produce them. Now, having found these autoantibodies in patients, we then tested the possibility that these autoantibodies could actually neutralize the interferon. Being an autoantibody against interferon means that the autoantibody does recognize the interferon, but it does not necessarily mean that the autoantibody can neutralize the interferon, that is, can block the activity of interferon. On the left-hand side, you see evidence that it is the case in vivo. We had serum samples from patients taken during the first days of hospitalization. And healthy controls don't have any measurable interferon in the blood, whereas patients with critical COVID-19 have very high or very low levels of interferon. And in red, you have the two groups of patients, patients with autoantibodies against type 1 interferon and patients with inborn errors of type 1 interferon. These two groups of patients have undetectable interferon in the blood, indicating for the genetic disorders that their variants in these genes prevent the production of optimal amounts of interferon. And in the case of patients with autoantibodies, that the autoantibodies actually neutralize the interferons in vivo. And serving as a control, you have that's the third group of patients here, the critical COVID patients without autoantibodies and without mutations in the genes controlling the production of interferon. So these autoantibodies neutralize interferon in vivo. And on the right-hand side, you have an experiment performed by Charlie Rice's team in which he attempted to test the possibility that these autoantibodies neutralize interferon alpha 2 in this experiment in an assay where the SARS-CoV-2 virus infects a cell line called HUH 7.5. The assay is relatively simple to understand. In the presence of interferon, these cells can control the virus. And serving as a control, you have here the blue line. This is blood or plasma from a healthy individual or a sick patient without autoantibodies. That is, the addition of this plasma doesn't change anything. The virus does not grow. That's the negative control. The positive control is the red monoclonal antibody. That's an antibody you can buy commercially. And you see that the addition of this antibody, even at high dilution, permits the growth of the virus. It neutralizes the recombinant interferon added in the experiment. And then all the shades of gray, these are plasma from patients. And you see that plasma from patients with autoantibodies beautifully neutralize the recombinant interferon, even at very high dilutions. And it actually works even better than the monoclonal antibody, attesting to the extraordinary power of these autoantibodies, their extreme neutralization capacity. So in short, what we've discovered is that in at least 10% of patients with life-threatening COVID-19 pneumonia, the disease is caused by pre-existing autoantibodies that neutralize at least 13 or 14 of the 17 type 1 interferons produced in response to the virus. It is, paradoxical or ironical that COVID-19 in these patients is in fact an autoimmune attack against the immune system itself, an autoimmune attack against intrinsic immunity. Now, the clinical implications are important. It is much easier to diagnose these autoantibodies than to sequence the genome. That can be done by ELISA in a couple of hours. Patients or individuals prior to infection known to have these autoantibodies are known to be at high risk upon infection with SARS-CoV-2. But there are therapeutic options. The first one is beta interferon. As you've heard, these autoantibodies neutralize the alphas and the omega, but they very rarely neutralize beta interferon, which is commercially available. Another possibility is plasmapheresis, which consists in depleting antibodies from the serum, which could be done perhaps together with, as Rick mentioned earlier, the injection of SARS-CoV-2 neutralizing monoclonal antibody. Now, what are the future plans? We have at least 10% of patients with the autoantibodies, but we now hypothesize, and we'll see if that is correct or not in the coming weeks or months, we hypothesize that there's perhaps a greater proportion of patients with autoantibodies against type 1 interferon, but autoantibodies that are in lower titers or that neutralize lower concentrations of interferon that may be sufficient to cause severe pneumonia. So that is in the pipeline, and we'll find out soon, I hope. So in fact, what we've discovered is that there is two mechanisms for the same mechanism. The pathogenic mechanism is insufficient type 1 interferon immunity in the first days of disease. But there are two mechanisms, at least, by which insufficient type 1 interferon activity is achieved: variants in genes that control its production, or antibodies, autoantibodies that neutralize its activity. I want to thank the many people who've contributed to this work. The first study was essentially the work of Chan Zhang, a pediatrician in the lab. I've had great help from Rick and Charlie Rice, also Michel Nussensvey. The second paper is the work of another pediatrician, Paul Bastard. Again, wonderful collaborators. And I want to thank Aurélie Coba and Laurent Abel in our lab in Paris, as well as Luigi Notarangelo and Helen Su at the NIAID. And last but not least, the people in the lab who've been so extraordinary during the last 6 months, our sponsors, the Fisher Center, the Howard Hughes, the NIH, Rockefeller, of course, St. Giles, and the institutions in Paris. Thank you very much for your attention.
Thank you, Jean-Laurent, for that incredible presentation. That was really amazing and exciting. We have a number of questions. I'll remind the audience that if you have a question, you can type it into the Q&A at the bottom of your screen. So to start off with the questioning, one question that comes from your work is whether the absence of appropriate type 1 interferons impairs the ability of these patients to make their own neutralizing antibodies. Is that a likely possibility, and is there any evidence regarding that?
So we have not compared the affinity of the antibody response to SARS-CoV-2 in patients with mutations in the genes controlling interferon, or by the way, in patients with autoantibodies against type 1 interferon versus the other patients. They have a positive serology, but we have not yet compared the titers, affinity, and so on.
Another question is, you've identified the interferon pathway as one obvious player in the susceptibility to severe infection. Do you suspect that this is the only major pathway, or do you think further genetic studies will turn up additional genes and pathways?
Well, I don't have an answer. My hypothesis is that it is a major circuit and probably not the only circuit.
In parallel, there have been studies looking at common variants that contribute to altered outcome of a COVID infection, and interested in your thoughts about the evidence emerging from those on at least chromosome 3 and ABO, but also one of the interferon receptors, I think, came up from one of those studies. And
right.
Your thoughts about that?
So in these studies, the odds ratios, or let's say the risk factors, are there, but they're relatively modest, maybe 1.3, 1.4. It is comparable, for example, with obesity or being a man as opposed to a woman. These are modest risk factors.
Yes, the odds ratio for your variants is infinite, as I noticed. You had 100 with the antibodies in the severely affected and 0 in the mildly affected. That's rather remarkable. So there was a question about math that came up asking if 0.1% of all cases are lethal with 220,000 deaths. How do you get to make the math work? Maybe the number is a little bit higher than 0.1%. Right, 1,000 times 220 would be 220 million people should be infected, and yet we only see a fraction of that.
Yes. The math is an estimation. The problem is we don't have an exact, we don't have the math. That is, we don't know exactly how many people have been infected. In part because some seronegative individuals we know have been infected. So we don't have the denominator. There's a big unknown. What's the denominator?
Yeah, I think it is fair to say that if you just took the assumption of at least 10% or at least 20% are infected, that would get you to about 3 in 1,000 at that level. And so we're in the ballpark, but there's certainly room for a better refinement of the estimate of frequency. And of course, it also depends on the quality of medical care. And as we all know here in New York and from experience in Italy, that the death rates were extremely high early on because it overwhelmed the medical capabilities to an extent, and we had no experience in managing these patients. So I think survival is significantly better now than it was then. And I hope we don't head back to the pandemic levels that ran through New York early on. Let's see, other questions coming in. So do you think it's possible that there will be direct therapeutic applications arising from the discovery of the interferon deficiency in these patients?
Yes, I think so. I can already tell you that last week, a couple of patients that I had followed for years with mutations in this circuit were infected with SARS-CoV-2, and I've treated both of them with PEG beta interferon the day they were diagnosed in order to prevent the complication. So I think for patients who have these variants in these genes, the treatment with beta interferon or alpha interferon is reasonable. In patients with autoantibodies, it is a more difficult matter to begin with because we don't know currently who has or who does not have these autoantibodies. But the test is being implemented in a variety of hospitals, and I think it will soon be possible to manage these patients better.
What do you make of the observation that the presence of these antibodies seems to be strongly correlated with age? Do you think we're losing tolerance with age, or how do you imagine this is coming about?
So the hypothesis we test now is that some of these patients may carry variants at birth on the X chromosome, and others may acquire somatic variants on the X chromosome. So we're testing, both the germline genetic and the somatic genetic X-linked hypothesis. That's the idea.
Yeah. And in terms of the risk for others, do you think there are other mechanisms for phenocopying interferon deficiency that might explain yet additional patients?
Well, Rick, what is really striking is that the patients with these inborn errors and the patients with autoantibodies are clinically indistinguishable from the other patients. And to me, that strongly suggests that this is the core mechanism at play in most and perhaps all patients. Insufficient interferon during the first 3-4 days. And then 10 days later, you have the cytokine storm, you have the hyperinflammation because interferons didn't do their job during the first 3-4 days. So I don't have evidence yet, but I like to think that this is a general mechanism.
Have you seen any explanation for racial disparities in COVID-19 from your genetic research thus far?
No, in the COVID pneumonia, we haven't. We have mutations in autoantibodies in people of various ancestries. There's no evidence that they're more prevalent in one group or another.
Any other associations with, it's striking that these patients appear to be well until they get infected with the virus. Would you expect other viruses that people would likely encounter in their lifetimes would cause severe disease?
Yes. So having found these patients with autoantibodies, we considered the possibility that patients in our cohort of with severe influenza or adverse reaction to yellow fever vaccine or other viral illnesses may be sick because of these autoantibodies. And the preliminary results are that it is the case. For the inborn errors, we already knew it was the case because these inborn errors of interferon could underlie, for example, severe flu. Now we know they can also underlie severe COVID-19, but the autoantibodies, their discovery begins with COVID-19, but now we're testing other viral illnesses And as I said, with exciting preliminary results.
As you might imagine, a number of people on the call have wondered whether there is going to be an assay available for people to get assayed for either the genetic variants or the antibodies to interferon.
Well, the genetic variants, the assay is already available because these genes are known human disease-causing gene. They're tested in most commercially available panels. And when a clinical exome or genome is being analyzed, they're already being studied. So anyone who's had his or her exome or genome sequenced has this information available. For the auto-antibodies, it is very simple to detect them. And I already know that a great many labs in the US in particular have developed this assay.
A couple of questions have come in asking about the relationship between interferon deficiency, cytokine storm, and its role in COVID pneumonia and the severe outcome of COVID pneumonia. The two seem somewhat antithetical, interferon deficiency and yet having cytokine storm as a mechanism for severe COVID-19.
Well, that's another great question, and it gives me the opportunity to try to explain what we now think is the model that really recapitulates the pathogenesis of COVID-19 pneumonia. People who have enough interferon activity during the first hours and days of infection control the virus well. The patients remain asymptomatic. They're not even patients. In the absence of interferon or with insufficient interferon activity, the virus is spreading in the lung, even through the bloodstream, reaching other organs. And then 8, 9, 10 days later, the virus has spread and leukocytes are attracted to infected tissue. And that results in hyperinflammation, including the cytokine storm. The cytokine storm is a storm of all cytokines except interferon because it was missing in the first place.
So there are additional questions about the relationship to severity. One of these is this question about blood type and risk of severe disease. Do you have thoughts on that?
Well, the risks, the odds ratios or the relative risk for the blood types are 1.2, 1.3. They're really modest. To try to give an idea to people attending this webinar, an odds ratio of, let's say, 1.5 means that if the risk in the population were, say, 1 in 15,000, well, then in people with the protective haplotype or genotype, the risk would be 1 in 10,000. That is, the diminution of the risk is very modest.
Several questions have come in about vaccination in the context of these antibodies to interferon. Do you think the absence of interferon response might impair the response to vaccination, particularly since these tend to occur in the elderly population?
Well, that's a good question. Again, I don't know. The prediction, though, is that it shouldn't have an impact. At least there's no reason to suspect an impact immunologically. Nevertheless, what I think these discoveries entail is that we want to be sure that the vaccination works in people with the autoantibodies and in people with inborn errors. They're the group to be protected.
Yeah, getting back to this fascinating observation of 95% of the patients with the antibodies being male. Would you elaborate further on your thoughts as to why this might be the case?
Okay, well, let's take a step back. There are we can think of three dichotomies. The first one is this can be a problem in the germline or a problem in somatic cells. That is, the problem can be present at birth or occur later in life. Second, it can be genetic. That is, the DNA material can be altered at birth or later, or it can be epigenetic. And 3, there are 2 sex chromosomes. It can be the X or the Y, or both, depending on the patient. So there are 3 dichotomies, and we're going to test them one by one. As I said, we're going to first test the hypothesis that the X chromosome is responsible and that it's genetic. and that it's genetic germline or genetic somatic. These are the first hypotheses we'll test. If this doesn't work, then we'll consider the possibility that the Y chromosome is involved, and that if that doesn't work either, then we'll try to test the possibility that these are epigenetic changes as opposed to genetic changes.
Well, I think the hour is getting late. Jean-Laurent, I want to thank you for first for the great science that you and your collaborative team have done. Second, for a beautiful presentation. And third, for so ably fielding all of the diverse questions that have come in. I'm sorry that we haven't gotten to answer all of the questions that have come in, but it's time to bring the discussion to a close. So thank you again to Jean-Laurent. And to all of you in the audience, thank you so much for joining us today. As you've heard, we still have a lot of work ahead of us, and philanthropic support remains vital to our progress. Please see the next slide for more information about how you might contribute and help us combat the COVID-19 pandemic. So until next time, all of us here at Rockefeller hope that you will stay safe and be healthy. Thank you very much for participating this afternoon, and I wish you all a very good evening.